start with a talk by Yanbin Yin, who's from Northern Illinois University, and he's going to talk to us about dbCAN family tools for automated CAZyme annotation of genomes and metagenomics.
All right. I want to first thank the organizer of this SIG for this opportunity to talk about my research on carbohydrate active enzyme annotation. So CAZymes is the short name of carbohydrate active enzymes. So I want to start give you a very brief introduction of carbohydrates. So for those of you who do not have a biochemistry background, so this is glucose. So glucose has 6 hydroxy group, and those hydroxy group are very high reactive, so they can polymerize into oligosaccharides. So by the way, glucose is a monosaccharide. So monosaccharides polymerize into oligo or polysaccharides with the help of glycosytransferases or GTs. So some of the polysaccharides like starches or cellulose are very well known because they are found in our food and also for bioenergy production. In addition to those saccharides, we also have hybrid molecule like glycoconjugates. So they have sugar group, but also other group like glycolipids or glycoproteins. So degradation of those carbohydrates or polysaccharides or glycoconjugates need a different kind of enzymes: glycosyhydrolysis, polysaccharidases, carbohydrate esterases, and enzyme with auxiliary activities. We also have those short modules which are non-catalytic carbohydrate binding modules, CBMs. So there's a very popular database called CAZyme developed by Bernard Harrisat in 1998. Basically classified all carbohydrate active enzyme CAZymes into these 6 classes, 360 different families. So all of the tools that I will be presenting today is based on this CAZyme database. So I want to mention that a lot of those CAZymes, they are modular, meaning that they have multiple functional domains. So as shown here in the bottom, we have 2 GH domains, which are catalytic. We also have 3 carbohydrate binding modules in the middle. So let me give you a background of the significance of carbohydrate metabolisms or CAZymes. So first of all, as I mentioned, that's glycosyhydrolysis, those enzymes are very important for bioenergy or biomaterial production. So second of all, carbohydrates are in our everyday diet. So not only that, they are also are used to feed our gut microbes. So we know that there are trillions of bacteria living in our guts. So those bacteria eat the dietary carbohydrates in our food. And also, if they don't have enough carbohydrates, so they will eat the host. So basically, human gut cell can secrete those glycoproteins called mucins. Those are also food of gut bacteria. So last thing is that microbial pathogens. So they synthesize and secrete a lot of carbohydrate active enzyme to degrade plant cell walls. So that's the major reason for plant disease. So here I show you a cartoon of the complex carbohydrates that are found in our everyday diet. As you see that, so if you eat a lot of vegetable and fruits, of course, you have a lot of carbohydrates in it. If you eat sushi, sushi has the seaweed wrapper, right? So that's basically algal-derived polysaccharides. So sushi also have rice, meat, so those are glycoproteins and starches. And mushroom, of course, that's fungus polysaccharides. Milk has glycoproteins, etc. So if we zoom in the plant-derived carbohydrates in our diet, as you see here, there's a very high diversity of carbohydrates in plant-derived food, dietary elements. And the point I want to make is that there are so many different kinds of glycosidic bonds that need to be degraded to break down so that you can release those sugars. So human genome only encodes 17 glycoside hydrolysis. So we basically rely on our gut microbe to fully utilize those complex carbohydrates. So shown here, I said that human has only 17 GH enzyme, but for example, those bacteria from Bacteroides phyla, so they encode up to 260 different glycoside hydrolysis. So you need to consider those bacteria, their genome size is only 1 to 1,000. They're basically 3-fold smaller than human genome, but the percentage of CAZyme encoding genes in their genome can go up to 8%. So that's why recent research shows that changing the dietary carbohydrates, changing the composition, will affect human health. So my interest since 2012 is how do you annotate newly sequenced genome, metagenome for those CAZymes. So I mentioned the CAZy database. CAZy database basically classify carbohydrate active enzyme into protein families and protein classes. However, they do not allow user to annotate newly sequenced genome for CAZymes. So that's why in 2012, I developed a database, basically a database of hidden marker models. Each hidden marker model represents one carbohydrate active enzymes. So we also have an online server to allow users submit their protein sequence to annotate genomes. So I mentioned that a lot of those CAZymes are modular. So with those domain-based signature domain hidden marker model, so we can very easily to annotate those modular proteins. So let me talk about the dbCAN hidden marker model. How do we build that? So first of all, we start with the CAZy annotated protein. In this case, family X has 4 proteins. And the reason that they belong to one same family, they must share some common domain. In this case, we scanned all the 4 proteins for some functional domain by searching against CDD of NCBI. And in this case, the red domain is found in all the members. So those will be extracted at the signature. So in addition to CDD search, we also did a lot of literature readings, especially for those short carbohydrate binding modules. So basically, we extract those signature modules or domains, and they will build multiple sequence alignment, we build hidden marker model. So basically, dbCAN is a hidden marker model database with 360 HMM. So you might ask, why not just use PFAM or CDD? So the reason is that only 30% of 360 CAZyme family are included in PFAM or CDD. And second reason is that their model is not trained on CAZymes because those PFAM or CDD are general protein family database. So as I said, that with those 360 hidden marker models, we can build a server so that user can submit their protein sequence to for CAZyme annotation, automatic CAZyme annotation. So the result page include a table, also a graphical display of the modular domain structure. So after our paper published in 2012 in NR's Web Server Special Issue, it received a lot of job submission. In last year, we received almost 20,000 job submission. That's just the web summation, not considering people who actually download our model and run them locally on their computer. So the top visiting institutions include DOE's National Lab, USDA, which is not surprising because those institutions produce the largest number of microbial genome, plant genomes that are related to bioenergy, biofuel research. Since dbCAN paper got published, there are 2 other methods came out for CAZyme annotation. The first one is diamond versus CAZy. So diamonds, I guess most of you have heard of that. That's equivalent to BLAST P and BLAST X, but much faster, 20,000 times faster. So now we can actually take a fully sequenced genome to do diamond search against full-length CAZyme protein annotated by CAZy already. So the other method published in 2017 is called PPR, which is short chimer motifs that are conserved in one CAZyme family. So basically, dbCAN and PPR make the same assumption. However, dbCAN use consecutive longer domain. However, PPR use conserved k-mer motifs. So HotPep is just a tool to scan a new protein for the presence of those conserved k-mers. So now, with the 3 different automatic tools, we can combine their results, and so that's why we have this dbCAN2 meta server. So in order to evaluate the performance of the 3 tools, we download some pre-annotated CAZyme from the CAZy database, and we try to figure out what is the best parsing threshold for each of the method. So we found for Hammer and dbCAN, there are 2 important thresholds. One is eValue, the other is coverage, how much of the domain is covered in the search. The second one, Diamond, basically just eValue, and HotPep has 2 parameters. One is frequency of those k-mer motif, and the second one is how many hits with that k-mer motif. So we tried a number of different thresholds and found those are the best threshold. And those best thresholds, we calculate the F-score for the 3 methods. So, so basically, Diamond is works the best for prokaryotes and worst for eukaryotes, and HotPep is the opposite. So Hammer plus dbCAN actually give you a more balanced F-score and slightly better overall performance. So more importantly, when we combine results predicted by at least 2 tools, we actually were able to improve the F-score from 88, 86 to 93 or 92. So basically, by combining results from 3 tools, we can improve the prediction accuracy. So in terms of speed, Hammer is the slowest, and Diamond and HotPep both are very fast. However, as I mentioned, dbCAN is built on those signature domains, so it has the obvious advantage, which is can be used to annotate multi-domain proteins, especially when you have multiple CBM repeats. We can report those repeats and also their locations. But the other 2 methods is very difficult to get those information. All right. So in addition to the integration of the 3 methods for automatic CAZyme annotation, we also have 3 other new features. The second one is that we combine results for better visualization, which I will show you in a minute. The third thing is that we allow DNA summation. So in our first version of dbCAN, we only allow protein sequence summation. Now we allow you to submit draft DNA genome or metagenome. So for that, we will predict genes first, protein coding genes first, and then do the CAZyme annotation. So the last new feature is that now we are able to predict what we call them CAZyme gene cluster, or CGCs. So we have a Python program called CGC Finder for that, so which I will talk about now. So I believe most of you in this room know about gene neighborhood. That's the so alignment-free, basically, alignment-free methods for gene function prediction. So there are all kinds of different alignment-free methods. So here we particularly look at this gene neighborhood, which is one of the guild by association method. So basically, based on literature, biologists showed evidence that CAZyme often work with other proteins like transporters, transcription factors, or signaling proteins to efficiently or effectively degrade complex carbohydrates. And those proteins that are encoding genes tend to be clustered in the genome and form operons or form gene cluster. And we name those gene clusters or operon as CGCs. So in experimental study, they actually call CGC as PUL, polysaccharide utilization loci. So here's one example. So this is the starch utilization system characterized by Jeff Gordon's lab in 2006. So shown on the top, there are a gene cluster or PUL with 8 genes. The first gene actually encodes a regulator, regulate the expression of all the genes. And if we look at the protein product, so DEF are actually signaling protein. They're used to bind different starch polysaccharide, and G is the GH enzyme, that's the CAZyme, to cut long polymer into oligomer, and C is encode a transporter, send those oligomer into the periplasm space. So where you have two other glycoside hydrolysis, A and B, further cut those oligomer into monomers. So now you have monosaccharides, which are transported into the cell with other transporters. So really, so this becomes a general mechanism for bacteria to degrade all kinds of complex carbohydrates. So in the past 10 years, there are over 3 dozens of POLs has been experimentally characterized in different bacteria from different environments. So here we see that POL characterized in insect gut, in cow rumen, in human gut, also in marine sediments, in soils, etc. Interestingly, all of those bacteria, they are from 2 major bacteria phyla, which is Bacteroides and Proteobacteria. And the other interesting thing is that shown in yellow and purple. So all those POLs, it has 2 important proteins. So one is the SUS-C, the other is SUS-D, which I showed you in the previous slides. So, so those experimental study, as I showed you, that there are a few dozens of them. However, if we are able to automatically scan all the fully sequenced bacterial genome to use bioinformatics, present all the predicted CDC online, that will be very useful for the community. So here we developed this CDC Finder as a Python program. So the algorithm is very simple. So first we, in addition to CAZyme annotation, we also annotate a genome or metagenome for the other 2 classes of signature genes, transcription factor TFs and transporter TCs. So second step is that we locate those 3 signature gene classes in the genome and then extend to both sides in order to include more signature genes. So here we have 2 important parameters. First one is the signature gene class. So the stringent one, of course, is to have all the 3 signature gene classes present in the gene cluster. The second parameter is when do you stop? Okay, I'm almost done. When you extend to both sides, you want to include non-signature genes shown here as gray box. So basically, the most stringent threshold is that you don't allow any. So with all those new features, we built this dbCAN2 meta server. This is the job submission page. User can submit protein or nucleotide, as I mentioned. They can choose submit genome or metagenome. So result page shows a Venn diagram overlap between the results of the 3 methods. There are 5 tabs. So I just want to highlight the CDC that we predicted. So we click on CDC tab, any of the CDC, we have this gene cluster diagram to show the member genes and also a table. So all those data can be downloaded to the user's computer. So last, I want to talk about this new database called dbCAN-seq, which is a pre-computed CAZyme sequence annotation, because we have all the 3 tools, and we basically scanned all the fully sequenced bacteria for CDCs for CAZymes. So here I just want to show you a basic statistic. So because we have over 5,000 of bacteria scanned, so here we showed that some bacteria phyla, like Bacteroides, Rucum microbiome, so those are major bacteria phyla found in human gut. They have the highest percentage of CAZyme encoded in their genome, almost 4% up to 10%. The other 2, Accidobacteria is found in soil, Thermatoga is found in hot spring. So those are environments that has a lot of plant or algal carbohydrates. So in terms of percentage of CAZyme located in CDC, so this basically evaluates the tendency of CAZyme form gene clusters. So we use a very stringent parameter, distance less than 1, and all 3 signature classes have to be found in the CDC. So basically, we identify 26,000 of CDC in over 80% of the genome that we scanned. And so here again, we compare different bacterial phyla in terms of the CDC percentage, and we found Firmicutes, Proteobacteria, which are 2 other gut bacteria found in human gut and found in cow rumen, has the higher percentage of CAZyme, clustered into CDC. So we are actually continuing this research to answer more biology questions. So last, I want to thank students and collaborators involved in this project. So we have undergraduate students, we have graduate students, we have computer science students and biology students. Actually, one of the main developer of the dbCAN 2 website is computer science undergraduate students. And the funding is from NSF and NIH. We have the website. If you're interested, you can go look at our website. Thank you. If you have any questions.
Okay, thank you. I think in the interest of time, we should move on. If you have any questions for Yanbin, you can ask them in the comment.
Okay. So we also have a poster.
There's a poster
Yes.
as well.